Yeah, now it's recording. So what are the factors, as we said, what are the factors that important for regulation of enzymes or affect on the enzymes work? Just remind you, by the enzymes, we said enzymes are proteins or biological catalysts. We called it biological catalysts, and these biological catalysts are used to speed up the chemical reactions. So they are made of proteins. We are talking about enzymes, okay? Enzymes made of proteins. They speed up
Reaction.
the reactions, yes, by what? Lowering,
lowering the activation energy.
yeah,
Reactivation energy.
the activation, the
Very
activation
specific.
energy, yes. And we said enzymes are
Enzymes is specific.
specific. What does mean specific? Means that enzymes work, yeah,
Suitable.
yeah, specific substrates, right? Substrates, specific substrates can attach on the active site of the enzyme. Then we said enzymes named by adding ASE to the substrate name, okay? And most of enzymes, actually most of enzymes named like this way. And last, we say the enzymes are not used up during, yes, during the reaction. Okay, it can be used in many reactions. We have factors affect, sorry, we have factors affect on the enzymes, affect the enzymes work. What are these factors? The main three main factors: number one, pH; number two, temperature; and number four, the surface area. Time. Let's move on one by one and understand what are these factors and how it affects on the enzymes work. First of all, regulation of enzyme activity. Enzymes can be affected by any variable that influences the chemical reaction, okay? And by the way, yani, we have more than these factors can affect on the enzymes work, okay? But yani, we are focusing on these. Enzymes work best at certain optimum pH value. At certain pH. This is called optimum pH. Means that the pH where the enzymes work in the best. And yesterday we explained what do you mean by enzymes work. The enzymes work means that converting the reactants into products in faster time, right? Okay, this is the enzyme work, speed up the chemical reaction. What does it mean? Means that converting the reactants into product in faster time, okay? So the enzymes work that under optimum temperature, under the suitable pH, okay? Any change in this optimum pH, yeah, affect on the enzymes work, okay? Does my voice clear for you guys?
Yeah.
Yeah,
It's
good,
clear.
Yes.
good. Many enzymes are affected by change in temperature. So each enzyme has optimum pH, and enzymes are affected by change of temperature, okay? But we should understand how exactly. Cells can regulate the activities of enzymes. Oh, look. Now, the chemical reactions in the cell not supposed to be on every time, right? Chemical reactions in the cell can be switched off or switched on in the cell. And the cell can use the enzymes as a key to switch on or switch off the chemical reactions. So if the cell wants this chemical reaction to be switched on and going on, so it give it this enzyme or switch on the enzyme or give the enzyme optimum pH and optimum temperature. Got it? What if the cell does not want to do this chemical reaction, does not want this chemical reaction to be on? So directly the cell what switch off the enzyme or stop the enzyme's production. So the chemical reaction will not work. Got it?
Won't be enough.
Yeah. So the enzymes, the cells regulate its activities by the enzymes, using the enzymes as a key to switch on or switch off the chemical reactions. Because of course, not all the chemical reactions are working in the same time. Got it? Yeah. Sometimes the cell need to store the sugars. So it needs the enzymes which are used to convert the monosaccharides into polysaccharides. Sometimes the cell need to do the opposite. It needs energy, so it needs to break down the stored starch into monosaccharides to use it to get energy, right? So in this time, what does it do? Huh? It switch off the enzymes which using for synthesis of polysaccharides and switch on the enzymes which are used to break down the polysaccharides.
Dehydration synthesis.
Yeah, you got it? You got the point? Easy thing.
Okay.
Most cells contain proteins that help to turn key enzymes on and off. So the enzyme itself has a key, like lock, let's say, okay, lock and key. It's switch off and switch on. We can switch on the enzyme itself and switch off the enzyme itself. So the enzyme has two forms: active enzyme, inactive enzyme. Okay, so if the cell need this chemical reaction to be on, simply just switch on the enzyme which is responsible or in charge on this reaction, or make it active enzyme. And the opposite is correct also. If need to stop the chemical reaction, so easy, make the enzyme inactive. Okay, got it?
Yeah.
That's good. Enzymes play essential role in regulating chemical pathway,
Mr. You're lagging.
making material. I'm lagging.
No, Mr. It's his internet.
It's okay.
Maybe
Okay.
it's his internet. Yeah.
Enzymes rules. We said this: regulating chemical pathways, making materials that cell needs, releasing energy. These are the chemical reactions examples, and these are the like releasing energy. This chemical reaction controlled by enzymes, transferring information, and other stuff. Let's see what are the factors that affect on the enzymes work. We said the temperature, pH, and the third surface area, but not written in the PowerPoint. Temperature, increase in temperature, increase the work of enzyme. Why? Understand, focus on this to explain for you. But do you have any question? Yeah, I see raised hand. Yes. Who raised his hand can speak.
Mister, I have a question.
Yeah, wait, Diego, yes, you don't need to raise your hand, Diego. Speak anytime. Yes, Diego.
What are the materials in the cell?
The
Like it says,
yeah,
the materials in the cell.
material cell is like university, is filled with things. We're gonna talk about it in the next chapter. The parts of the cell and the components of the cell. But you
Mr.
know,
Basim.
there are thousands of chemical reactions take place in the cell, and all of it controlled by the enzymes. Okay, and the cell control these chemical reactions, the thousands of chemical reactions, by the enzymes, by switch on or activate the enzyme or inactivate the enzyme. You see? Yeah.
Okay.
Yeah.
Mr. Basim,
Yes, Muhammad.
you explained for us 4 roles for the enzymes. For the enzymes, are there
Yeah.
more?
Yeah, of course, but yani, here we need the main essential enzymes role in the cell.
Or the
Okay?
4 main roles.
Yeah, main.
Basically.
The main, yes, the
Can
main.
you show the roles again?
Sorry?
Can you show the roles again, the essential roles?
Yeah, I will come back to it, but I want to explain first the wait, I want me to repeat the roles.
Yes.
Yeah.
Oh, okay. I'm back to it. Yeah. Enzymes essential roles. The main rules in the cell. First, regulating chemical pathways. We said that it speed up the chemical reaction and regulate the chemical reaction. What does it mean? Chemical pathway, guys. Some reactions, like for example, respiration. You know respiration, right?
Yes.
Respiration is a breakdown of glucose or the sugars in the mitochondria to release the ATP, the energy. And you know it is C6H12O6 plus oxygen give off H2O plus CO2 plus ATP, the energy, right? Actually, what you're supposed to know, and you will take this, do you see this equation? This
Yes.
chemical equation, we have chapter talk about it. Why chapter talk about it? Because it's not only one chemical reaction. Actually, this equation, what you have, what do you see now? It has hundreds of chemical reactions. Okay? Or tens of chemical reactions. So we going to study it. And what you're supposed to know that we have many of enzymes regulate this chemical reaction, the respiration. So that's why we called it chemical pathway, not chemical reaction, actually. This one is equation of chemical pathway. Pathway means a series of chemical reactions. Got it? When we say
Yeah.
chemical
Yes.
pathway, a series of chemical reactions, but they have to be ordered and arranged. How they ordered and arranged? By the enzymes. The enzymes regulate which reaction and what is the next reaction. Got it?
Yes.
Yeah,
Makes
like if we have 5 students, we give them 5 duties, but these duties have to be arranged, ordered. Okay, so one by one, we say first Diego, then Muhammad, then Ali. So by turn, okay. Ali not supposed to work before Diego because break the order in this way, right? They have to be in order. Same idea here. The enzymes
What
work in order.
happens if you break the order?
No,
What happens?
so the chemical pathway will be damaged. You will not get the result. Okay. So
Okay.
here, regulating chemical pathways, making materials that cells need. Of course, the enzymes work on the production of materials which are needed. Releasing energy. I just said the respiration pathway. Transferring information. Enzymes are important, for example, to copy the message which found on the DNA. And we're gonna study this in details. I will remind you by that we have many enzymes work on that. When we study how the DNA, is responsible on building up protein. The DNA send message to the parts of the cell which are responsible.
Translation.
Yeah, which are responsible on building or synthesis of protein. It's sent it, or the message is written by enzymes. Enzymes write the message. The message, by the way, is RNA, mRNA. We're gonna talk about this in details. Okay, so the idea that the enzymes are used to write the messages to build up the protein. Okay, let's go to the factors. I was talking about temperature, the first factor. Guys, scientists said that any chemicals to react together, any chemicals to react together, they need to what? Collide to each other. They need to collide to each other
Have
in
enough energy.
and enough energy, excellent, to react and what? And change to product and change to product.
To
Here,
exchange bonds.
yeah, so here moving the molecules of the substance is important. What we call the movement energy, what we called it? Huh?
Thermal energy. Thermal
Kinetic energy.
No,
Kinetic
kinetic.
energy.
Excellent. Kinetic energy, right? The kinetic energy is important to help the molecules to collide to each other. So they react, that speed up the reaction or allow
So
the
what about thermal?
allow the, wait, just a second. The kinetic energy is the movement energy. Okay, allow the molecules to collide to each other. Okay, collide to each other. But what increase the kinetic energy? Yeah, Abu Ghandar, you just
Thermal
said it.
energy.
Yeah,
Thermal energy.
yeah, heat, right?
Thermal.
Yeah, so heat or the thermal energy allow to increase the kinetic energy. More heat or more temperature, you
More
know,
movement energy.
temperature is measurement of amount of heat, right? More temperature means what?
It's an average.
More
More
kinetic.
More kinetic energy.
Excellent. More temperature, more kinetic energy. So that's what more enzymes work. And this time, look, if you put enzymes, also the molecules need kinetic energy to collide with the enzymes and attach on the active site. Got it? To attach on the active site. So,
Yes.
yeah. So here the kinetic energy is important too for what? For binding the substrates on the active site of the enzyme. Got it? So more kinetic, more temperature, more kinetic energy, so more formation of enzyme substrate complex, right? So that increase the work of enzymes, right? Right?
Yes.
Good.
So they'll finish in less time.
Yeah. So more.
Yes.
But is there limit? Is there limit for this temperature?
Yes.
Yes.
Because the hydrogen bonds.
Why there's a limit? Raise hand.
Because
Raise hand if you have answer. Why there's a limit for this temperature? Why we don't keep raising the temperature up more and more so that increase the work of enzymes, because more collagen between the enzymes and the substrates. Yes, yeah, I see only 3 guys, 4 guys raise their hands, and the others, alhamdulillah, do not share with us. I don't know why. Anyway, let's see. Tijan. Yes, Tijan.
So the enzymes will start to deteriorate and destroy themselves because the hydrogen bonds will start to break. And
Yeah.
so that's why too much heat is
Thank you very much. Thank you very much for this. Yeah, that's correct. Actually, you know, enzymes are proteins, and we talked before that it's a, by the way, it's tertiary protein, and we talked the tertiary protein that is maintained by hydrogen bonds, by SS bonds, you remember, by hydrophobic bonds, or yeah, hydrophobic bonds.
so what is SS bonds?
Sulfur bond. Okay.
Oh, okay.
But you didn't explain
These are the main bonds which maintain. These are bonds maintain the 3D protein, right? such as enzyme. Raising temperature in the beginning is okay, does not affect on the enzyme structure. So that increase the kinetic energy and allow binding of substrate on the active sites. But up to limit, more than this limit, what will happen for the enzyme? The enzyme will be denatured. Will be what? Denatured.
Nature.
That means the bonds will
Denatured means
Hey.
damage. What does it mean, denature? It means
It will
losing the
destroy.
shape, losing the shape. Look, the shape of active site changed. Because you know this active site is formed by the formation of the enzyme, 3D structure, by the bonds. Break down the
Sebastian,
bonds. The excess heat, or let's say the extreme heat, the extreme temperature, break down the bonds in the enzymes, so that lead to denature the enzyme, damage the shape of enzyme. Got it? So in this case, okay, it increased the kinetic energy of molecules, increase the kinetic energy of molecules, but it's in the same moment it's break down the structure of enzyme. So how to draw the graph? It's like that. This is the optimum temperature. Optimum temperature, it's the best temperature where the best kinetic energy for the enzymes work. But after this optimum temperature, the enzyme is denatured, right? So how to draw it like that? See? That means at these temperatures, the enzymes still work. So the enzymes still working in low temperature. This is low temperature. Okay? This is here extreme temperature. Okay? At low temperature, the enzymes are still working, but very low activity. Here, enzymes activity. Okay? The enzymes activity at low temperature is very low, but at the optimum temperature, it's what? It's the best. The best.
It's the best for it.
Yeah.
Sebastian,
Just a
how
second.
do you find the
Just a second. After the optimum temperature, the enzymes work slow down until it's what? Denature at this temperature. Okay, it's the extreme temperature, it's denature. Here we say the enzyme denature. Yes, questions?
Sir, how do you find the optimum temperature? How do you find it?
Yeah, by
Yeah,
experiments. By experiments. The scientists did experiments for the enzymes at its optimum temperature. For example, all the enzymes, most of enzymes in your body, inside your body, work at which optimum? Guess, which optimum temperature?
37.
32
37.
degrees
Body
Celsius.
temperature.
Yeah, thank you.
Body
Actually, it's 40.
2
At 40.
pH.
At 40, because if it's 37, so if your body temperature raise up to 40, you're supposed to die because all the enzymes in your body denature, right? But actually no, but it's still dangerous, right? Yeah. The enzymes in your body optimum temperature is 40, is around 40. Okay. Okay, guys. So all the enzymes in the body temperature, all the enzymes in the body work at optimum temperature about 40 degrees. Okay.
Approximately 40.
But in different animals, they have different optimum temperatures. Okay, different organisms have enzymes with different temperatures. Okay.
Okay.
And that's why
Yes.
for example, fruits in the plants grow in spring. The plant, for example, making new fresh leaves in spring. The leaves fall down in fall season. All of this due to hormones and enzymes. Okay, the enzymes.
Okay.
Yeah. Now, farmers, for example, by experience, they know what are the best months to grow the seeds. planting seeds. They
5
do not,
minutes left.
yeah, planting seeds, not in any time. No, there's specific times for every type of seeds, okay? That's due to the temperature.
Season.
Yeah, the season, the temperature, okay? For the enzymes. Yeah.
Mister,
Yes, yes, Ali.
so when you get sick, your body's temperature gets higher.
Yeah,
Does anything
yeah.
enzyme?
Actually, when you get sick, your body temperature, look, the optimum temperature 40. It's 37. It's like in safe side. The enzymes work in safe side, but not on the optimum. The optimum 40, the best. Now, at 40 is still optimum, by the way, and it's between 37 to 40. It's optimum. Now, more than 40, what will happen for your body enzymes?
Die.
Not the nature directly. Yeah, the nature little bit. Yani the nature of the enzyme is
So
not
like
suddenly.
slowly. They
Yeah,
just slowly denature.
slow down,
Yeah.
then
Nature.
until it's the nature here,
Dead.
the nature. Okay, so
Mister,
40, wait, 45, 44, these are extreme temperature where human dies if the body temperature raised to this temperature, 45, 44, because
chance
yeah,
dying.
or at least, yani, if not die, they lose some functions of body organs, like lose brain function, or lose heart function, or lose um,
Liver
yani,
function.
yeah, nerve functions, okay?
Memory loss.
Yeah, because of enzymes denature, okay?
So should we join again? 3 minutes left.
Sorry?
3 minutes left.
Yeah,
3
I know.
minutes left. Should we rejoin?
I know. No, we not joined because we lost about 5 minutes in the beginning because of the Zoom application. Yes, Muhammad Walid. Okay, let me finish the second point.
Okay.
The pH, very easy thing, the pH. You know, pH is the concentration
Potential of hydrogen.
of hydrogen. And we know, we just said, the protein, the 3D structure protein such as enzyme, is controlled by hydrogen bonds. So of course, the hydrogen concentration affect on the hydrogen bonds. Huh? Any change in the pH directly denature the enzyme. Okay? So every enzyme has optimum pH. Okay, here the enzyme activity. Okay, what is the enzyme activity on at the optimum pH? It's on the highest. The enzyme working. What if the pH change? Hop, denature. Okay, so the enzymes denature, or here
Miss,
denature.
is there a pH level greater than like there's a scale of the pH level from 0 to 14.
From 0 to 14, yes.
Yeah, but can it increase the pH scale like to 15 or 16?
No, of course,
Possibly.
we discussed this before, no. So guys, the optimum pH is the pH where the enzymes work on the best. Any change in this pH directly, the enzymes denature. Look, there's a difference here in temperature. In temperature, at low temperature, it still works, okay? Because kinetic energy. But in pH, no. Lowering pH, increasing pH, denature. We have enzymes in your stomach. It works at 2 pH.
2.
Yeah. So any change in this pH, the enzymes in your stomach stop working. In your small intestine, your enzymes work at 8 pH. So is it possible that the stomach enzyme to work in small intestine?
No, it's impossible.
No,
No.
impossible.
No.
Yes, it
No.
will die.
No.
It will die.
Denature.
The
Denature.
nature.
Right? Denature. Okay? Okay. Your blood pH.
It's 7.
7.4.
7.4
7.4.
pH.
Yeah.
7.4. Enzymes work at this pH. Okay? These are some essential examples in your body. That's it for today.
Bye.
Okay.
Inshallah, we
Mr.
have
Bassem.
lab session, inshallah. If not this week, next week, okay, inshallah, during the lab session, we will work on enzymes. Okay, inshallah. That's it for today. Thank you for joining.
Mister,
Enjoy
when
your
are we going to
day.
do anatomy?
Mr. Bassem,
Anatomy.
When we study the body, yes. Quickly, the time is
I
over,
have
huh?
a question.
Quickly.
Okay, goodbye, mister.
Quickly, let him ask. Yes, ask.
Sir, I have a question. For
Ask.
yesterday, when you said that's the username password, I try to log in, but it keeps asking for location.
Let's discuss this in the group. Let's discuss in the group, okay?
Mister,
Show the video,
Yeah,
video
Mr.
and password.
On the group.
Just
Okay?
send it, Mr.
On the group.
Okay.